All right, hey everybody. Thanks for joining us for the Thursday seminar. It's my pleasure to introduce our speaker today, Dr. Andrew Rassweiler from Florida State University. So before I do the introduction, I'll just get some of the little housekeeping stuff out of the way. Right. Remember, all audience participants will be muted throughout the seminar. Do not attempt to turn on your video or share your screen during the talk. And at the end, there'll be an opportunity for the audience to ask live questions. So once the speaker finishes, right, then you can use the raise your hand feature in Zoom to notify the meeting host that you'd like to ask the speaker a question. Then we'll call on you and we'll allow you to unmute yourself and ask your question for the speaker. Then after the question session, anyone's welcome to stick around and we'll have a more informal chat with our seminar speaker. So with that, as I said, yes, I'm very pleased to introduce our seminar speaker today, Dr. Andrew Rassweiler from Florida State University. And I may slip up and call him Rass. So that's because we have known each other for over 25 years now. And that's basically what everyone calls him because Rassweiler is way too long of a name to say. And so, yeah, and Rass, a bit about his background. He is a quantitative marine ecologist, and he does a really excellent combination of, field work, manipulations, observations, monitoring, and ecological modeling. So I think you'll hear about a whole variety of those things in his talk. Yeah, so as I said, we've known each other for 25 years now. We did our undergraduate together at Princeton University. We played on the water polo team together as freshmen. Our junior spring, we spent the whole semester together down in Panama for this Princeton ecology program down in Panama with 14 other students. Rass then was really excited to start working on coral reefs, so he did some undergraduate research where he was studying the effects of scavenging by moray eels down in Curacao in the Caribbean. And we also overlapped down there where we worked for a graduate student, Stuart Standen, who's now a professor at Scripps, and helping him with his graduate research. Well, then we both went to graduate school together at UC Santa Barbara. And so while Rass was there, he was working with the Santa Barbara Long-Term Ecological Research Group. His advisors were Russ Schmidt and Sally Holbrook, and he worked very closely with Dan Reed and others involved in that program. He did some really cool, just sort of side projects, helping, looking at kelp forest dynamics and measuring primary productivity and net primary production in kelp and looking at the drivers of those patterns. He then did his PhD work where he was studying alternate stable states and phase shifts in kelp forests, but he wasn't studying the typical one we think of, shifts between kelp forests and urchin barrens, but instead was studying shifts between kelp forests and these pachythione, these small sea cucumber barrens, and what factors led to shifts from a kelp forest to a barren, and then shifts back from that sea cucumber barren to a kelp forest habitat. He then, like many of us, knowing that Santa Barbara is such a lovely place to live, refused to leave for a while after he graduated. And so he did his postdoc and then kind of a research sciences position there, where he continued to do a lot of work studying kelp forest dynamics over time. He started doing a lot of really cool work looking at sort of resource and fisheries management issues, and then also studying the effects of MPAs. And that's where we have one publication together where we looked at the long-term changes in the Channel Islands Marine Reserve Network. And then I guess it's been the past 4 or 5 years now, he's been a professor at Florida State University. His first graduate student, Scott Miller, was one of my master's students. And he's been doing a lot of work now studying coral reefs, particularly in Moorea. And he's been there looking at the effects of disturbance and fishing and how that's been affecting grazing behavior and the dynamics of coral reefs. And I think that's partly what we're going to hear about today. And so with that, Rass, we'll turn it over to you. And yeah, thanks for coming to give this seminar for us.
Thanks, Scott. That was a great introduction and really, wow, we have a long history together. That's fun though. And I appreciate your mentioning Scott Miller because he will figure pretty prominently in this story, and we'll come to some of the stuff that he's leading partway through. But I'll just get started. Thanks everyone for coming. I really appreciate the invitation to come tell you a little bit about my work. I'm an applied ecologist and I work in a pretty wide range of marine ecosystems. So that includes tropical coral reefs, temperate rocky reefs, and shallow seagrass habitats. And in general, I'm interested in interactions between ecosystem state and human activities, but not just how humans impact ecosystems, which is pretty classic conservation biology, but also how changes in ecosystems lead to changes in human behavior and the resulting feedbacks and consequences for the dynamics of the ecosystem. So in the past few years, my work's been split across 4 major topics. I study feedbacks between fishing and resilience in coral reef systems. I've been working on developing better methods of measuring marine biodiversity, and that work's been mostly in kelp forest ecosystems. I have some continuing work on spatial fishery management, and I've been studying spatial patterns of seagrass-associated ecosystem services along the Gulf Coast of Florida. But today I'm just going to highlight some recent research in 2 of these areas. I'm going to spend most of my time on the first section. I'm going to tell you about a project I have studying the resilience of coral reef systems and how feedbacks between fishing and ecosystem state affect their dynamics. And then I'll show you some work focused on spatial fishery management, exploring how climate change is likely to alter the effectiveness of networks of marine protected areas. So coral reefs are in decline around the world. This change has been more dramatic in some areas than others, for example, in the Caribbean, but the declines have been taking place worldwide. And one of the most important questions in coral reef ecology is what makes coral reefs resilient. So these habitats are exposed to periodic disturbances which cause the loss of coral. And these include physical disturbances such as storms and marine heat waves, and also biological disturbances such as outbreaks of coral-eating crown-of-thorns sea stars. Now, historically, corals have recovered after such disturbances, but increasingly we're observing reefs around the world failing to recover, and instead we're seeing other community states, particularly those dominated by fleshy macroalgae, taking over and persisting. But the variation in these outcomes is a crucial puzzle for ecologists. Why does coral bounce back in some locations and decline in others? So I'm going to tell you about a project I have trying to understand the role of fishing in driving shifts from coral to algal dominance in the lagoons of Moorea, French Polynesia. So this is a picture of Moorea, just to give you a sense of it. The island is right next to Tahiti in the center of the South Pacific. And most of the research I'm going to tell you about today is focused on Moorea's lagoons. So these are these light turquoise areas around the island. They're between the reef crest, so that's this white line here, and then the shore. The lagoons are mostly shallow with a mix of coral and sandy habitats and lots of fish. So if you go and swim around in Moorea's lagoons right now, you would see a patchwork of ecosystem states. So some locations have beautiful high coral cover, while other locations are basically solid beds of macroalgae on every hard surface. And these macroalgal dominated states are principally made up of turbinaria, which is a pretty nasty algae. It's protected both physically and chemically from herbivores. Now, recent experiments in Moorea's lagoons have demonstrated that the macroalgae dominated state represents an alternative stable state, or at least an alternative persistent state. So if you take a patch of hard substrate and you remove the macroalgae, so if you scrub it right down to the rock, the action of herbivores will maintain that algal-free state for years, even as the neighboring chunks of habitat, which you did not manipulate, stay dominated by macroalgae. So it's hard for the algae to get established, but once it takes over, there are positive feedbacks that make that transition really hard to reverse. And we know that herbivorous fish in this system are key for maintaining the algal-free state. Because if you put a cage on a spot that doesn't have algae, it'll very rapidly be taken over. So excluding herbivores rapidly leads to macroalgal dominance. So we have these interactions between the benthic community, like between corals and algae competing for space, and then that competition is mediated by fish herbivores. But while herbivores are a key driver of benthic outcomes, herbivore abundance is itself strongly influenced by fishing. So overharvesting of herbivorous fish has been implicated in many shifts from coral to algal dominance around the world. And often it's told as a very simple story of a causal chain from people down to benthic dynamics. So people remove the herbivores, which releases the algae to overgrow coral. But there's actually considerable potential for feedback loops in the system, both within the biology, but also between the ecological system and the social system. So to make progress on the key ecological questions, such as why some locations have been taken over by algae while others retain coral, we need to consider these human activities. Now, it happens that we know relatively little about fishing practices in this system. So in the first part of the talk, I'm going to tell you about some work that my lab has been doing trying to understand the fishers' foraging model, how they make decisions about what to catch and where. And then I'll come back to integrating that with the ecology to understand the dynamics of the system. So I'm going to address 3 questions about fishing. First, the most basic is like what sizes and species of fish are being harvested in the system. And as I mentioned, we know that herbivores are crucial for resilience, so it's important to understand how heavily targeted they are. Then I'm going to explore how the catch changes as the relative abundance of fishable species shifts. And finally, I'm going to ask how fishers change their activities as other non-fished components of the ecosystem change. So to address these questions, we wanted to quantify what fish are being caught. And in Moorea, much of the catch is sold by the roadside. So people display their fish for sale, hanging in strings from racks. And I should mention this is primarily a spear gun based fishery. So people go out spearing fish, then they come back and they sell them by the roadside. And these racks are really convenient for us because we can take a photo of the rack and distinguish each individual fish being sold. So we now have run these surveys for a bunch of years with students in my lab identifying the fish being sold and estimating the length and weight of each fish based on comparison with a sizing bar in the photograph. So that's this thing right here. So we have a ton of data on the catch. And to look at the relationship between the reef ecosystem and fishing behavior, we paired that data with ecological surveys of the fish on the reef. So I got this data from 2 sources, a US-based project, the Moorea LTER, and from a French-led monitoring project. And each project involved counting and sizing fish on transects in different habitats. So here's a breakdown of the size distribution that we see in the catch. That's this top graph up here, and the distribution we see on the reef. That's this bottom graph. And you can see immediately that fish in the catch are much larger than those on the reef. So the top graph is shifted out there to the right. And in particular, fishers seem to ignore fish smaller than 15 centimeters. So this is aggregated across all species, but the same patterns hold if you look at the species individually. Now, if you look at the taxa in the catch, the one thing that stands out is the prevalence of herbivorous fish at the top of the list. So I highlighted here all the herbivores in green, and more than 50% of the catch is made up of herbivorous taxa, particularly unicorn fish and parrotfish. But we wanted to know how the catch compares to the ecological community. So what does this tell us about rates of harvest mortality on different taxa on the reef? And I should mention here that I only kept fish above 15 centimeters for this comparison. So this over here is all fish of all individuals of fishable size on the reef. And we find that most things that are common in the catch are also common on the reef, but the opposite is not true. So many taxa that are common in the on the reef are rare in the catch. And I highlighted 2 dramatic examples, Tenocatus and Zebrasoma here, which together make almost 20% of the fishable sized biomass on the reef, but are rarely targeted by fishers. So this suggests that these species are largely exempt from fishery mortality in this ecosystem. So the answer to the first question is pretty clear. Fishers are heavily targeting herbivorous fish and are very selective both in size and species targeted. They prefer to catch larger fish and they target some species while avoiding others. So let's talk about this second question about how selectivity changes over time. So there have been some substantial shifts in fish species composition on Moorea's reefs over the period we have data. So big shifts in the ecological system. How do fishers change what they harvest as the fish community changes? Put another way, this is asking if human impacts on the reef are changing in a predictable way over time. So I'll sketch out a couple of our hypotheses. Going into this project, some locals had told us that there were these traditional responses to ecological change. And so people would change where they fished and what species they targeted as the ecosystem changed. So we wanted to document those traditional rules and then think about how they interacted with ecosystem state. And I was very hot on this idea in the beginning of the project, that these traditional rules would prove to be some sort of secret to ecosystem resilience. But we had students do a ton of surveys on the island, and going around and asking people about traditional fishing practices, and we basically got nothing. So if this was once a feature of fishery management in Moorea, it's not only not being practiced, it's mostly on its way to being forgotten at this point. Okay, so a second hypothesis is that which fish get harvested is basically set by demand. So people like to eat some species more than others, and spearfishers have a choice about which fish they shoot. So this might suggest that they're not going to be very responsive to changes in the reef. It would predict that fish are going to show up in the catch at the same ratios, even if their relative abundance on the reef is changing, because the catch is really driven by people's preferences. And this hypothesis was consistent with our surveys. So 80% of the people that we talked to said they didn't change anything about what fish they caught, even as the ecosystem changed. But an alternative hypothesis is that the catch is mostly determined by supply. So if the biomass of a species goes down on the reef, it'll go down in the catch. One that becomes more abundant on the reef will rise in the catch. And this is basically what we see, at least for the common species. So we see that fishing is set by supply. So for example, this data is for the most abundant genus in the catch. This is unicorn fish. And on the y-axis is the proportion of this taxa in the catch. And on the x-axis is the proportion of the fishable biomass on the reef made up of unicorn fish. And you can see that there's a really tight match here. So basically each step up or down in abundance on the reef was perfectly reflected in the catch. And this same relationship holds for most of the common taxa, although not with such an incredibly high correlation. So from an ecological perspective, this means we can assume that the proportion of individuals of each taxa, which are being removed by fishing, is relatively constant over time. Like the per capita rate of harvest mortality is not going to swing up or down as the fish population moves, as long as the total fishing effort stays the same. Okay, so we found a lot of fishing on herbivores. We found selectivity across sizes and species, and we found that per capita mortality rates are consistent even as the abundance of fish changes. Now I want to move to thinking about how fishers might be responding to ecosystem change beyond changes in the fishable resources. And in particular, I want to ask how fishers might respond to increases in macroalgae. So essentially, I've been telling you about feedbacks here from fish to human behavior. And now I'm going to talk about feedbacks directly from the benthic ecosystem, and specifically changes in coral and macroalgal cover. So we tend to think of algal-dominated state as degraded, and thus we might intuitively think that a loss of coral would be bad for fishers. And I think that most coral reef ecologists would agree that fishing will suffer over a long enough time scale. But it's not actually obvious what happens on shorter time scales when coral's been lost, but where most of the coral-associated structure is still present. So the physical structure of the reef is still there. So we tend to think of, so just to give you an example, here is data from a recent analysis we did of ecological community states in the Caribbean. And the bars here compare the value of the fish community in locations with high, medium, or low coral cover around the Caribbean. So this upper bar is high coral cover, medium coral cover, and low coral cover, and the y-axis is basically the dollar value of the fish, to the fishery. And so there's a ton of variability in the fish community within each category, but basically no meaningful effect of coral cover on the potential value of fish found on the reef. So I mentioned this just to highlight that I didn't have a strong a priori hypothesis for whether fishers might prefer, avoid, or even just ignore shifts from coral to macroalgal dominance. So I took a bunch of photos of lagoon habitats in Moorea, trying to get pretty similar physical context, but to capture photos with a wide range of macroalgal cover. So here's some examples. Some of these photos have very little algae, while some have a ton of macroalgae. So that's what you're looking at, like up here or over here is basically like a whole forest of little stalks of turbinaria. And then I had a student interview a bunch of fishers, and what he would do was he would deal out 6 photos from this set to each fisher, and he would ask them to rank the photos in order of where they would most prefer to fish for various species. So I quantified the organisms and substrates in each photo, and I used the photo rankings to ask 2 questions. First, I assumed that fishers would prefer locations with more high relief structure. So locations that were more physically complex as opposed to flatter locations. Many reef fish are associated with physical structure, and I wanted to control for that possibility. And then my second question, sort of my real question, was once we control for the presence of structure in the photograph, will there be any relationship between the degree of macroalgal coverage and the ranking that each photograph receives? So here are the results with the effects of high relief structure on the left and the effects of macroalgal dominance on the right. And I estimated the effect sizes of these factors jointly with linear models, and I plot them with 95% confidence intervals. So each point is the estimate of the effect of a particular on a particular group of fish, with the groups ordered so the ones most targeted by fishers are at the top. And the result is very consistent for 4 out of the 5 taxonomic groups. And in particular, it was consistent for parrotfish, unicornfish, and surgeonfish. Those are these circled groups here. And those together make up about 65% of all herbivorous biomass on the reef and more than 90% of fished herbivorous biomass. So for these species, fishers preferred locations with more high relief substrate, so more big chunks of structure, but they showed an aversion to photos with more macroalgae. Okay, so I've talked a bunch about fishing at this point, and now I'm going to come around to exploring what these results mean for the dynamics of the ecosystem. So I'm doing all this work on fishing because I really want to understand patterns of coral and algal dominance in the lagoons. I want to understand why patches of algae form and persist where they do, but also why the whole system hasn't been taken over by macroalgae at this point. And I'm going to talk a lot about resilience in this next section. And by resilience, I just mean the capacity of the coral-dominated state to resist disturbance or recover from disturbance. So effectively, the ability of coral to outcompete macroalgae. So as my starting point for this next section, I used an existing model of interactions in Morea, which describes how coral and macroalgae compete at small spatial scales. So as I mentioned earlier in the talk, we know that the system has positive feedbacks, which means that algae has trouble taking over if it's rare at first, but once it becomes established, it can persist. And one detail of the biology of the system, which has the potential to explain this phenomena, is that algae are very resistant to herbivory in their adult phase, but much more vulnerable as juveniles. So if the adult algae can reach some critical density, they can take over the system. But if algae are rare, the little juveniles get gobbled up before they can reach adult size. So I took this benthic model and then I added a model of the dynamics of herbivorous fish and a model of fishing. So the fishers are harvesting the fish are consuming the vulnerable macroalgae. The vulnerable macroalgae are growing into adult invulnerable macroalgae, which are competing with corals for free space. Now, this model gives the potential for alternative stable states. So here's a plot where I put fishing pressure on the x-axis and equilibrium cover of coral on the y-axis. And if fishing pressure is low, the coral will be abundant. So that's like down here. If fishing pressure is very high, the coral is basically going to disappear because the macroalgae is going to take over because the herbivores all are removed. And then there's an intermediate level of fishing at which corals can persist if they're initially dominant. But if coral is reduced and macroalgae is allowed to take over, it'll prevent corals from recovering. So that's in here. And in that area where the 2 lines are separated, the outcome depends on the initial state of the system. And we know from experimental results that the lagoon in Moorea is currently in this intermediate range of fishing and herbivory. So what happens in this model if we add a tendency for people to fish less if macroalgae becomes more abundant, as was suggested by my survey results? So the top graph is the result I just showed you, and the bottom is a version where people fish less if a location is taken over by macroalgae. And within a particular location, this behavior is good for coral resilience. So basically the behavior is pushing these lines to the right. It means that coral remains dominant at higher levels of fishing pressure, and it somewhat narrows the range of fishing pressure at which either coral or macroalgal state could persist. But looking only at the dynamics within a patch misses important processes. So if fishers are leaving the patch because it has too much macroalgae, then that fishing effort is likely being displaced into other habitats around the island. So I developed a spatially explicit model of like an idealized island where I have patches, sort of a ring of linked lagoon patches around an island. And within each patch, all the local ecological dynamics are occurring. So coral is competing with algae, herbivores are eating the algae, and people are harvesting the herbivores. But these patches are connected. So herbivores can disperse between neighboring patches, and fishers can choose any location around the island to target. And by default, I assume the fishers basically just go where the herbivores are, so they distribute themselves in proportion to fish biomass. But I also explore the possibility that they're avoiding macroalgae. And I'm particularly interested in cases where the ecosystem state varies around the island. So I simulated disturbances with spatially variable coral loss, in which some patches lose most of their coral and other patches are less impacted. And as my measure of resilience, I ask what fraction of the patches recover to coral dominance after a disturbance. So I looked at this effect, the effect of this behavior across a broad range of scenarios. So in this plot, disturbance severity goes from left to right. So disturbances get more severe as you move over towards the right side of this graph. And the line of results just along the top of the graph basically represents the scenario where fishers are ignoring macroalgae, so they're not responding at all to changes in macroalgal cover. But as you move down this graph, you're looking at scenarios in which people have a stronger and stronger aversion to fishing in macroalgal patches. And if we want to know how this behavior is affecting resilience, we compare outcomes at the top of the plot to outcomes farther down in the plot. And so you can see in a broad region on the left side of the plot, basically this behavior is helping coral. So if fishers ignore algae, most of the patches recover coral, but some remain trapped in a macroalgal dominated state. So that's this sort of greenish area up here. If fishers avoid macroalgae, this creates coherent dynamics across the lagoon. So coral recovers everywhere instead of only recovering in some patches. So this is this bright yellow area. This is all like full coral recovery here. Then there's a region over on the right side of the graph where the behavior basically doesn't matter. If the disturbance is severe enough, you'll lose coral everywhere, no matter what the fishers are doing. So that's this dark blue area over on the right side here. But then there's also this narrow range of severity in which the behavior can actually hurt coral. So if fishers ignore macroalgae, some coral would recover. So that's this light blue area at the top. But if fishers avoid macroalgae, then the tendency to synchronize dynamics across the lagoon can actually lead to collapse everywhere. So again, that's this dark blue area down here. So these results confirm that fishing can affect coral resilience, but they also reveal the importance of feedbacks with human behavior, and they highlight some key gaps in our knowledge. In particular, I'd like to know how strongly the fishers avoid macroalgae. I showed my photo sorting exercise, which tells us something about the direction of the preference, but really not very much about the strength. And there are also other key factors we don't know. So how far are fishers willing to travel, and what other aspects of the ecosystem are they responding to? Now, as an ecologist, I want to understand why coral has persisted in some locations and declined in others, both in Moorea and also around the world. And the work I just showed you suggests that we can't answer those ecological questions without understanding how fishing is interacting with the system. But fishing is only one of many factors influencing community state on coral reefs. Fish are moving around, they're growing on the reefs. So how much does spatial variation in fishing effort really affect the fish communities? And how important are top-down effects when we know that there are also lots of other factors that affect benthic dynamics, such as things like nutrient loading? And the problem is that it's really hard to answer these questions with existing data. The relevant processes are occurring on spatial scales of hundreds to thousands of meters. We have some ecological data in the system, but it's pretty small scale. So for example, there's this long-term ecological research project there. They do annual sampling of 6 sites. They spend a huge amount of work and they do a great job sampling those sites, but it's a pretty small window into the dynamics of the system, particularly if you want to know about these spatial processes. Now, the traditional solution to this is basically to use models to scale up from small scale observations to make predictions about larger scale dynamics. And that's what I've shown you so far. But what I really want to be able to do is to address some of these questions directly with data on the correct spatial scales. And that requires coming up with new methods of collecting data in the ecosystem. And that's something that I've been working on. So for example, I have an anthropology postdoc who is actually living down in Moorea, and he's been able to recruit a bunch of fishers who've been tracking their fishing using smartphones. So in this top panel here, these yellow lines are all GPS tracks, which we can use to figure out exactly how much time people spend fishing in each habitat. And then the other cool thing is that at the end of each fishing trip, the fishers have been taking pictures of the fish that they catch and sending them to us. So I have a team of undergrads who've been analyzing these photos, basically identifying and counting the fish and measuring their size. So I now have a ton of fishing data on the scale of the whole island, mapped right down to where on the reef the fish were caught. But I also need ecological data of similar scope. So for the past 3 years, we've been conducting really large scale surveys across the lagoons. And when I say we, I mainly mean my PhD students, Scott Miller and Ali Dubell, pictured here looking heroic. Scott is a proud Moss Landing alumni out of Dr. Hamilton's lab, so I'm pleased to tell you a little bit about what he's been up to. And at this point, Scott and Ali have surveyed about 200,000 square meters of reef habitat. So each of these little dots on the map, they're all piled on top of each other, but each is about a 25 square meter segment of habitat that they've surveyed. And the basic approach is to swim along, counting herbivores and fish larger than 10 centimeters. And the observer tows an inner tube behind them with a GPS that's synchronized to their watch. And we keep track of the fish observed in 1-minute increments. So that means we can map fish onto the reef in areas of about 25 square meters. So here's an admittedly staged photograph I took of Ali counting fish. And below it is a map of, the blue lines here are a bunch of the GPS tracks. And down in the bottom is a zoomed in version where you can actually see the segments there, the individual transect, 1-minute transect segments there. But in a way, counting fish is the easy part. The challenge is also to describe the benthic community state in these areas without slowing down. So we want to know about the coral, the cover of coral and macroalgae, and we also want to know about the physical characteristics of the locations where we're seeing the fish. And so to do that, we basically just strapped a GoPro onto the bottom of the inner tube with the GPS facing down. And so the GoPro is passively taking photographs of whatever's under it, 90 shots per 1-minute transect. And when I dreamed this plan up, I thought maybe 10 or 20% of the photos might be usable, and it would be just enough to give us a sense of what the habitats looked like that we were moving through. However, it worked way better than I expected. So here's some examples of the photographs that what they look like. And the good news is that in shallow water, so shallower than about 5 meters, the photographs are almost all usable. The bad news is that this means that I have about 1 million photographs of shallow habitats in Moorea linked to my fish counts. So that's a lot of photos to look at. And so I assembled a team of undergrads, and along with Scott and Ali, we looked at thousands of these photos using a program called CoralNet. And CoralNet is a system which is designed to let you do point contact. So you identify the organisms under a set of randomly placed points, and then that lets you get estimates of the percent cover of various organisms and substrates. Now, the cool thing about CoralNet is that it lets us train a computer vision algorithm to identify points automatically without a person having to look at them. And after doing about 2,000 photographs with the undergrads, the computer vision system got pretty good. So my PhD student, Scott Miller, is just finishing a manuscript describing this whole process and validating both the fish counting technique and the computer vision system. And here are some results, basically comparing the accuracy of the computer vision approach to a more traditional approach where humans score each photograph by hand. So let me take a moment to walk you through what you're looking at. Each of these rows is one of the things that we identify. So either a substrate like sand or an organism, such as a particular genus of coral or macroalgae. And the farther to the right each dot is, the more accurately this approach can describe variation in the category at the scale of a 1-minute transect segment. So about 25 square meters of reef. So a point all the way on the right side of this graph means the approach is 100% reliable at describing spatial variation in the organism or substrate. A point on the left side of the graph means the approach is basically useless. And I've ordered the categories from most common at the top to least common at the bottom. And what you can see is that we're doing a pretty good job for most of the common categories. Each red dot is the result with humans using 8 randomly selected photos to describe the cover of a category. And the light blue dots show the accuracy of the computer vision system, also using 8 images. And the humans beat the computer for most categories, like 70% of the categories, but the computer is pretty close. The computer does lousy on some of the rarer categories down at the bottom, but the humans don't do great either at describing variation in these rare categories. But one of the advantages to the computer is that there's no reason to stop at 8 photographs. So here I've added those dark blue dots, and those represent the accuracy of the computer vision approach if we give it 64 photographs to work with. So again, the results are pretty close for the common categories, but now humans are winning only 2 of the 17 cases. So we're pretty excited about this approach as a scalable way to describe reef community states. And the word scalable is important here. Given the amount of data we've collected so far, it would take about 3,000 person hours to score 8 photos from each transect segment that we've counted fish on. To do the same analysis with the computer vision system will take us less than 5% of that time, including all of the time we spent training the classifier. And importantly, the marginal cost is very minimal, so we can keep collecting data without creating huge chunks of new work for ourselves. So the system has incredible potential to really let us scale up our ecological observations. Now, Scott's been working with this data to try to tease apart the importance of different drivers of fish community structure. We're interested in the importance of fishing, but also the influence of physical structure and aspects of the benthic community, such as coral and macroalgal cover. Scott and I have also been collaborating with another research group, which has been measuring patterns of nutrient loading across Moorea's lagoons. So we're trying to use this data to estimate the relative importance of top-down effects, such as fishing, versus bottom-up effects, such as nutrient loading on the benthic community. My new PhD student, Ali Dubell, collected a lot of this data as a technician, but over the last semester, she's been working on linking the ecological data with the spatial fishing data that I mentioned at the beginning. Basically doing a much more sophisticated job of understanding the Fisher's foraging model. And this is like very much a work in progress, but she's finding that the fishers are less likely to go to places with a lot of sand and also less likely to go to places with a lot of macroalgae. So the survey results I showed you before seem to match where people actually go fishing on the reef. Now, one of the fun things about collecting so much data is we're able to use it for various side projects that weren't even in our mind when designing the survey. So for example, one of my undergrads, Alyssa Turner, wrote her honors thesis using this data. When helping us analyze the photos, she got interested in bite marks she was observing on the corals, and she went back to quantify bite marks on a ton of corals. So this is one without bites, and this is one with a ton of bites. And she was able to show that, to link this up to our ecological data and show that corals had higher risk of being bitten when there were more parrotfish, but also there was a dilution of individual risk in areas with more live coral cover. Now, there we go. At the moment, one downside of this approach is that I have humans towing the cameras. So the surveys end up being where the people swim, and it's hard to come back and replicate a survey precisely in a later year. So I'd really like to be able to characterize these habitats over multiple years so I can describe how spatial patterns change over time and how the community at particular locations changes. So we've been experimenting with these homebrew autonomous vehicles, basically bolted together from PVC and auto nav parts developed for fixed wing drones. And they work really impressively so far. I'm planning to roll these robots out in Moorea this summer and maybe in the Caribbean as well. So I just want to wrap up this section by returning to the big picture motivation for the work in Moorea. I want to better understand what leads to coral reef resilience, both to advance the ecology, but also to help guide better management of the reefs. There's an existing network of marine protected areas in Moorea, which is universally acknowledged to be a failure. But while everyone agrees it's not working, there's no consensus on what should replace it. So we've been working closely with local stakeholders in our research, and I think there's a real window of opportunity to improve management in the system. Okay, so with that, I'm going to leave Moorea and just spend a little time telling you about another project I have focused on the spatial management of marine resources. So in the early 2000s, I worked in the MLPA process, so helping to design California's marine protected areas. I got involved halfway through the process, so I'm not responsible for the Central Coast protected areas, but I worked on the South Coast and the North Coast reserves. And as most of you probably know, a marine reserve is just a chunk of the ocean in which fishing is restricted or prohibited. And the main goal of these areas is often conservation, but they're also increasingly being used as a fishery management tool. And in a series of papers, I was able to show the potential value of marine reserves for improving fishery outcomes, so increasing things like fishery profits and fishery yields. And I found it was particularly important to account for patterns of dispersal when designing marine reserves. So I'll just illustrate that with a cartoon. Many coastal fish produce larvae that disperse long distances, riding ocean currents for weeks before settling and growing to become adults in a new location. And so it turns out the details of ocean currents are really important for these populations. A fish in one location might be very valuable for the next generation because larvae produced there will settle in other nearby habitats. Fish in another location might be destined to have most of their offspring washed out to sea away from any suitable habitat. And if you focus your reserves on locations which are good sources of reproductive output, you can increase fishery yields. But one thing that's always worried me about these results is that you don't get improvements just placing marine reserves anywhere. So here's an example where the black dotted line represents the best outcome possible with non-spatial management. So just limiting fishing effort, but letting fishers go wherever they want. Now the red line represents the outcome from a perfectly designed reserve network that takes dispersal into account. And so you can get 10% higher yields from putting reserves in the right places. But then the gray histogram is the distribution of possible outcomes if you were to design marine reserves randomly. So protecting the same total area as in the optimal reserve network, but randomizing the placement. And you can see most of those random designs result in negative fishery outcomes, so a loss of yield. So of course, one of the conclusions here is just, well, you should do a good job when designing reserves. But there's a more subtle problem. What if you do a good job of placing your marine reserves today, but conditions are changing over time. Is your network going to continue working, or do you need to constantly redesign it as the environment changes? So I've started exploring these questions in the context of climate change. And in particular, I've been thinking about how climate change affects larval dispersal. Now, this is a convenient place to start because we have very specific and well-supported predictions for how dispersal will change in a warming ocean. So across a whole range of organisms, it's been observed that increasing temperatures lead to more rapid development. And that means that marine organisms will spend less time in their larval phase and will not be able to disperse as far under future climate conditions. Perhaps surprisingly, it also suggests that survival in the larval phase should go up. So the larvae are growing faster and spending less time at vulnerable sizes, so a larger fraction should reach their destination. So I'll show you some results based on some spatially explicit bioeconomic models I developed of fish species in Southern California. I won't go into any details of the model, but each model includes data on the habitat distribution of the species, details of its life history, and estimates of dispersal probabilities based on modeling of ocean currents. And I incorporate these into spatially explicit models of fish populations and models of fleet dynamics in the region. And in these results, I'm focusing on how the placement of marine reserves affects total fishery yield. So I'll give a little more detail about how we're estimating dispersal probabilities, because this is the main factor that'll vary under future climate scenarios. I use the output of a regional ocean modeling system to estimate the probability that larvae produced at one site are delivered to any other site in the region. So this is based on the pelagic larval duration, so the amount of time that the larvae of a species spends in the ocean currents, as well as the season in which the species reproduces. So to simulate future connectivity, I assume a 2.5-degree warming in the coastal ocean. So this was based on a 2100 scenario from RCP 8.5. And I calculated the change in the pelagic larval duration and larval survival for each species following O'Connor et al. 2007. And the output of the regional ocean modeling system predicts new patterns of connectivity for each species based on this shorter larval duration. So just to give you an example of how connectivity changes, we can consider kelp rockfish. Its pelagic larval duration today is about 60 days, and we predict in a warmer ocean at the end of the century, its larval duration would be about 49 days. So it'll spend about 20% less time dispersing. And this increases the median connectivity between patches by about 10%, but dispersal probabilities between some patches are doubled and others are unchanged or even slightly less connected. So patterns of connectivity change. How important are these changes for the optimal network of marine reserves? So to examine that question, I optimize the placement of marine reserves under current dispersal and then compare that to a network of reserves which are adjusted to be the best possible network under future larval dispersal patterns. And the result is a pretty substantial change in reserve placement. So redesigning today's reserves for future connectivity would involve removing some reserves and adding other new reserves for each species. So on average, about 70% of today's reserves would be removed, and about 20% of the reserves in the optimal future network would be new. So pretty big change in the network design. So I've shown you that if connectivity changes, the optimal locations of reserves changes. And we know that poorly placed reserves can be ineffective or even counterproductive for fishery management. So does that mean that it's basically a waste of time trying to strategically design marine reserves? If we design them to work well today, are they going to be ineffective in the future as the climate changes? Fortunately, the answer seems to be no. This is the value of marine reserves designed for today's conditions, but operating under the climate predicted at the end of the century. So each bar represents the increase in yield that the marine reserves would provide relative to having no reserves. So they're all above the line, they're all positive relative to no reserves. So this suggests strategically designed reserves will still work well in the future. Now, it's true that redesigned reserves that have been tweaked for future conditions will do even better. So those are the darker bars here for each species. The unmodified reserves would, or sort of the modified reserves would yield 32% higher yield. Oh, sorry, the unmodified reserves yield 32% higher yields than no reserves. The redesigned reserves yield 38% higher reserves on average. So it's a little better, but not like dramatically better. And you might think, well, this is just because marine reserves are good. Any reserves will work out well in the future. I'll just show you that's not true. So these gold bars here represent outcomes from randomly placed reserves, which cover the same area as the optimal reserve design. And you can see randomly placed reserves actually reduce yield by about 15% on average across the species. So I went into this project worried that strategic designs might be pretty fragile to changes in dispersal patterns, and I was surprised by how robust they are. The results suggest that investing in good design up front can pay dividends even with pretty substantial changes to connectivity. But there are many other factors, effects of climate change that I didn't look at here. So there's a whole list of other things that are likely to change. I think of this very much as a first step at the problem. And I'm doing more work on this, including more comprehensive predictions about the effects of climate change on marine organisms, models that operate over larger spatial scales and incorporate both ecological and evolutionary processes, and analyses that consider a wider range of management interventions than just marine reserves. So I'll just wrap up by returning to my research themes. I told you about my work on coral reef resilience. I'm really excited about some of the progress that I've made, and in particular about the potential to study subtidal ecological processes on larger spatial scales. So when ecological communities change, often what's actually happening is that one community type is spreading across the landscape. And that's a process which is really hard to study with most existing marine biological data. I also told you about some modeling work I've done on fishery management under climate change. Figuring out how to design management to work well under climate change is sadly likely to be increasingly crucial in all ecosystems. And these kind of analyses can help us determine what management tools to use. I didn't get to talk to you about marine biodiversity or seagrass ecosystem services, but I'm happy to chat with students about those topics at happy hour or at another time. And again, I really appreciate your inviting me to come present, and thank you for coming to my talk.
All right, well done, Dr. Rassweiler. Thanks so much, that was exciting. And man, your graduate students must be in the best swimming shape ever, swimming around the entire island of Moorea. So yeah, we are open for questions. Raise your hand if you would like, and we will call on you. I guess while waiting for others, I will ask a question. So I was kind of curious thinking about the model, right, that you were that you used to kind of simulate those alternate stable states in Moorea, and the main link between the herbivorous fish and what they were consuming was right those vulnerable turbinaria. But there was also a box down to the turf algae, right, which is another thing that they really like to eat as well. So do you think the model outcomes would be different if you also had them consuming that box, and maybe if there were different preferences, I guess, that kind of fit, yeah, kind of which one they consume more, the turbinaria or the turfs?
Yeah, that's interesting. The turf box at the moment, I'm not really modeling it explicitly. It's just kind of what I'm calling bare space. But the thing which I think you're getting at, which is on my list, is not the effects on the turf, but the effects on the parrotfish, right? So I don't have any feedbacks of food availability, like a location that has a lot of bare space presumably has better food quality, so it's going to have faster growing parrotfish. And I think I'm interested in what will happen with that, but I haven't gotten to putting it in yet.
Yeah, and it may attract more fish right into that area, and they
Totally.
just like the fishers avoid the macroalgae, right? The fish themselves may
Right.
be avoiding the macroalgae stands, and so yeah, I just thought there could be some cool feedbacks there depending on the interaction, but of course it makes it more complicated.
Yeah, it does, and that's also where I'm hoping that all of this spatial data we're collecting will unravel some of that for us, right? Because from the fisher preference, I don't know if they're saying they are avoiding macroalgae because they don't like macroalgae, or just because they like fish and they think there are going to be fewer fish when there's macroalgae around, right? So that's something that's sort of the whole point of this giant sampling campaign, is to sort of tease apart the different, what actually in the field is driving the fishing behavior.
Yeah, awesome. Awesome. All right, Travis has a question.
All right. Hi. Thank you for the talk. That was really interesting. I love this kind of the diversity of it all. So my question for you, and I hope that you're able to answer this. I found it pretty fascinating, this notion that a formerly good and productive MPA through environmental changes, it could become a poor functioning MPA. And I was wondering if you had found that there's a lot of pushback on this idea from whether it's any policymakers that you've dealt with, or just, I guess, regular citizens, because it just seems that it's hard to get people on board with these big policy changes, right? And if you're yanking them around, they can be kind of resistant to it.
No, that's an interesting question. I interpret this, I think, the opposite way that you frame it, though, Travis. To me, when I started this modeling with the climate change, I was expecting that actually the results I was expecting to come up with was I was expecting to come up with how often you had to redesign the marine protected areas in order to keep them working. I expected over 100-year period that my take-home message was going to be, do you need 3 redesigns, or do you need 5 redesigns, or can you get away with 1 redesign? And where I ended up was actually like, oh no, you don't even really need 1 redesign. Like they continue working. So it is true, if you were to fully re-optimize, you would take out a lot of existing MPAs and put in a lot of new MPAs. But my take-home message is really that it's more likely that you've put your marine protected areas in, you've been through the fight, you're not going to go back to that, you've got what you've got, and that actually seems to work pretty fine, at least for the sort of narrow, at least for the differences in dispersal, which I was focused on here.
Got it. Yeah, cool. Thank you very much for clarifying that.
All right, Emily has a question.
Hi, Andrew. So I know that when you were talking about the work that you were doing in Moorea and the coral reef resistance, and you just mentioned again when you were talking to Scott about how the whole point is you're trying to find the correlation between the fishing practices and the structure of the community. And you mentioned that you didn't know necessarily why the fishermen were kind of avoiding or showing a preference to non-macroalgal dominated communities. I was wondering if you guys did any type of like surveys of the fishermen themselves, or your postdoc that Scott, I think his name was, has he asked them specifically? Have you done surveys just asking them like, hey, why do you prefer this fishing spot over this one? Or are you just leaving it up to like the ecological data?
Yeah, that's totally good questions. The anthropology postdoc has been asking those kinds of questions, and it's a whole mix. If you ask people, do they prefer fishing in the algae or not, you get kind of a mixed result. You get about 80% people saying they don't like fishing in the algae, but then 10% of the people say they prefer fishing in the algae, which is like part of why I came up with this photo sorting exercise, because I was a little confused about what we were getting out of just the direct question. When you do ask them the direct question, a lot of them say they're just less fish in the algae, but the other thing that they say is that the fish are like harder, they like are sort of camouflaged in the algae, and they go in and out of the algae, so it's a little harder to spear them. So those are the most common things to say. But like some spear fishermen say, oh no, they like the algae because then they can hide in the algae, which seems like a little improbable to me. But so I think we're like trying to understand that, but we've gotten a little bit of a mix of answers on it.
Okay, that makes sense. I was just wondering if what they were telling you was correlating to the ecological data, but I
Oh,
understand
yeah,
it
it definitely runs in the same direction. So at this point, we have what they originally said. Most of them say they don't like the algae. Then we've got this photo sorting exercise, which says they don't like the algae. And we're just beginning to get like the GPS tracks where we're like, oh no, we went out and looked at what's there and there's algae there and they go fishing other places in the neighborhood, right? So that's like, from my perspective, that's kind of like the gold standard because that lets me quantify the strength of the aversion.
Right. Okay. Thank you. That makes sense.
I'll ask another question while we wait to see if there's any others. Do these fishermen spend any time fishing out on the forereef? I mean, all your work has kind of been the lagoonal habitat. So I'm kind of curious sort of what's happening with the fishing pressure out there, versus the dynamics of the reefs, compared to what's happening in the lagoons.
Yeah, it seems that from our surveys, it seems that about 70% of fishing effort is in the lagoons. About 10% is out on the forereef on the outside of the reef crest, and about 20% is in the passes. So there's a lot of fishing concentrated right in on the drop-offs at the passes. The forereef's just like a lot harder to fish in because it has to be kind of a nice day to be out there, and you need a more substantial boat and often someone to be driving the boat. And so there is a lot more fishing in the lagoons, and everything I showed you here has been like very focused on like, let's just think about the lagoon habitats. The other reason I've been focusing on the lagoon habitats is that the forereef habitat just doesn't have very much algae on it right now. Like the same experiments that show that the lagoons are in this alternate stable state kind of thing where either algae or bare space can persist for years. Those same experiments, if you do them on the forereef, it's like the algae all gets eaten, problem solved.
And so is the fish biomass, the herbivore biomass, way higher out there on the forereef if it's not that heavily fished, or is there some weird habitat stuff going on too?
Somewhat higher. There's also, so we get into we get into details here. The Turbinaria and the other macroalgae, like the most important herbivore is actually not parrotfish, it's Nasounicornis. And that's very heavily fished in the system. And there are definitely more of them and bigger ones out on the forereef. They're hard to count reliably because they know people are trying to kill them. And so they run away when you come nearby. So I would say we don't have great estimates of biomass of Naso.
Yeah, cool. Then my last question, right there. So there's a few of the herbivorous species that are common but not targeted, right? The Tinochetus, the Zebrosoma. Have you talked to the fishermen about why they don't? I mean, I know I've dissected some of them, and they're hindgut fermenters, and they're stinky. So, but if that's all they got, would they fish them too?
I've never asked. I don't think we've asked much about Zebrosoma. We've been really interested in the Tinochetus because it's so common on the reef, and it seems to be two things. One is ciguatera poisoning. So it's definitely a species that you can get poisoned from. And there are actually a few fishermen that do target Tinochetus, but it's only a couple guys. And it seems to be that there's a understanding that there are certain places in the lagoon that people feel like it's safe to eat them. But you kind of have to, you have to buy from a fisher that you trust knows where to go to get ones that aren't going to poison you.
Yeah, I guess they're more of a detritivore anyways, right? So maybe more likely to pick it up.
Yeah. Well, this is the thing. When you work with the anthropologists, like they would go on for 10 minutes here about like the rubbery flesh, and some of them, a lot of it comes down to taste also.
Awesome, right? Any other questions? All right, if not, anyone that wants to stick around, we'll transition to the happy hour and more informal chats, and you can chat more with Dr. Rassweiler. And let's thank him for his seminar. It was fantastic.